Well, we're in the home stretch, gang. Realize that there's one more regularly scheduled lab. Then we have Thanksgiving. Then you have your lab exam after Thanksgiving. Then you have, I think, 3 lab periods to work on your unknown. Remember that? So remember that Thursday we're going to have lab, the follow-up to Wednesday's lab. And then Friday we'll have the exam. So we're just flip-flopping periods. Okay? Okay, we've been talking about pathogens and diseases. And to wrap up this particular section, we need to say a few words about the study of epidemiology. Basically, epidemiology is concerned with causes, frequency, and distribution of diseases. In some respects, you can think of a medical, as an epidemiologist, as a kind of medical detective. A person who goes out there in the field or maybe does research in the lab, but is always asking questions like, who is being infected? Where are the infections occurring? What's causing the infection? Why do we have this particular outbreak? Okay? One of the things that is really important and is the basis of epidemiology is a very robust and systematic reporting mechanism where this particular information goes through a set of steps where physicians report to county health officers report to state health offices, state health offices report to the CDC, Center for Disease Control. Center for Disease Control shares their information with the World Health Organization. It's really important to have this sharing of information and make sure that everybody knows what's going on. And another basis of epidemiology is statistics, data. And one of the pieces of data, or one of the types of data that's particularly important, is the incidence and prevalence of disease. This particular graph right here shows you the incidence and prevalence of AIDS among U.S. adults from the period of 1986 to 2008. The incidence refers to the number of new cases. Prevalence refers to the total number of cases. But based upon this particular general information, there's a lot of strategies that can be developed to both try to prevent AIDS, and a big part of AIDS prevention is centered around education, and then also treatment programs. Okay, we might establish a vaccination program for a particular disease if we see that it's needed. We might recommend that if there's an outbreak of hepatitis associated with a particular restaurant, then we find out the people that visit that restaurant and tell them to go to their doctor and get a gamma globulin shot because you've been exposed to this particular virus. So this all kind of folds together under this particular field of epidemiology. As I said, a real key to epidemiology are the fact that certain diseases are considered significant enough that they are referred to as reportable diseases. If a disease is reportable, if a doctor comes across this, they must share that information up the chain, starting with their county health office. How many of you worked with Dr. Mormon when you did rotations at the hospital? Okay, he's our pediatrician. He's raised or been the doctor for most of the kids in town growing up, including my kids, and he is our county health officer. And then he interfaces with the state health, and then the CDC, etc. Let's go here. Here is a table showing the nationally notifiable or reportable infectious diseases currently by statute. Okay, so anytime a doctor runs across any of these kind of things, some of which we've talked about, we've mentioned cholera, for example, before. I know diphtheria is on here someplace, I believe. Tetanus is on here. So these are the kind of the most important diseases that doctors really have to notify. when the information gets to the CDC, the Center for Disease Control, they publish weekly the MMWR. That stands for the Mortality and Morbidity Weekly Report. And basically this is a summary nationally of kind of what's going on in infectious disease. The information might be tabular form, showing some particular information about particular disease and prevalence and incidence. It oftentimes is in graphical form, showing trends by year, by state, by age group. Possibly a breakdown by age group or sex, possibly by ethnicity. There's all kinds of ways you can parse this data when you have access to it to provide pictures of what's going on and what the trends are. For example, if you look back here at TB, what's something very interesting you notice about this data just pops out at you. More cases of males than females, right? Okay, and that begs a particular interesting question. Why? Why is that the case? What are the implications of that? What are the implications for education, for treatment? What's the basis? Some patterns start to emerge when you look at the incidence and prevalence of particular diseases. And there is some terminology associated with those particular patterns. One of the patterns is that diseases are oftentimes endemic. That means that they are found in a particular region, a particular geographic region, over a fairly extended period of time, in a fairly constant number of cases. So for example, here is the distribution of valley fever. And valley fever tends to be endemic to these southwestern states, Southern California, Arizona, New Mexico, Texas. If you looked at the distribution of Lyme disease, you would see clusters more up here in the far northeast. Although one of the interesting things that's been happening with Lyme disease is that it's spreading. And it's now becoming much more common in upper Midwest states like Indiana. Typically, a disease is endemic because the host or a vector of that disease is associated with a particular geographic environment. And one of the things that's happened with Lyme disease is that the range of the white-tailed deer, which is one of the main hosts for the tick that carries that disease, its range is extending, in part due to climate change. So, fairly constant number of cases, pretty stable, okay, particular geographic regions. Some diseases have a pattern which is considered to be sporadic. And they're just kind of a few cases here and there. No real discernible pattern. You might get a couple cases in this state and then not see it for a while. A couple cases in this state. Rabies is a good example of a disease that is typically sporadic. Probably one of the most misused words in epidemiology is the word epidemic. What does typically the term epidemic kind of convey to you? What do you think about it? We say a disease is epidemic. What do we usually think? Yeah, huge number of cases, just wiping everybody out. Okay, that's kind of the public's perception of the word epidemic. But that's not really what epidemic means. Epidemic really has nothing to do with the absolute number of cases. What it has to do with is the number of cases exceeding normal patterns and expectations. So we could say that rabies has become epidemic if there were several hundred cases in one state, because the normal pattern is a sporadic distribution where you might only have one or two or maybe no cases. Okay? So statistically, is it exceeding the norms that we normally observe? That is what epidemic refers to. What does pandemic mean? It means across continents. Okay? So this is an epidemic that is spread across continents. So we have talked about worldwide influenza pandemics, for example. But again, the key is not necessarily the absolute number of cases, but whether the numbers are exceeding what we normally would expect. There's all kinds of career possibilities within the field of epidemiology. There is descriptive, there is analytical, and there is experimental epidemiology. Descriptive epidemiology deals a lot with trying to find the location, the dates, the patients that are infected, and to look for patterns of infection. Oftentimes, what descriptive epidemiology starts looking for is to try to find that patient zero, that first case that is causing the outbreak or the infection. Probably the most famous example of descriptive epidemiology was John Snow's study in London of cholera, and tracing cholera back to a particular town pump that was contaminated with those particular bacteria that caused that particular disease. He did this literally by going door to door and knocking on doors and taking a census of who was sick, who wasn't sick, and then plotting that particular data on a map and looking for clusters and looking for patterns that might suggest the source of a particular infection. Analytical gets into more detail trying to determine the course or the mode of transmission or even possible prevention of diseases. And oftentimes when you get in analytical, you're kind of working backwards. You're finding where disease is located, possibly that initial patient zero, and then you're trying to analyze that individual and see what disease do they have, how do they get it, etc. So sometimes it's kind of a retroactive type of thing. Experimental evaluates hypothesis. Basically, this is where Koch's postulates really come into play, and trying to determine whether a particular disease is associated with a particular pathogen. A lot of that work in both the analytical and experimental epidemiology is carried out at the Centers for Disease Control. And that's where they have all those heavy biocontainment labs where they can work with all kinds of nasty bacteria and virus. And it's a pretty exciting career. It's also a dangerous career. Okay, working in that bio-level safety 4 environment. Some of you may know Dr. Chikwana is working on his degree in public health. And public health is really an outgrowth of epidemiology. And you can find all kinds of different careers in public health. We have the option now of a 3-2 program with IUPUI for both biology and chemistry majors to work on their degree in public health, kind of like a cooperative med tech program. But there's a lot of options there in a lot of different settings, from people who like to be out in the field working with individuals, to people who like to get into education, to people who want to do lab work, all kinds of opportunities there. And so that's really an emerging and important part of our whole medical system, because public health really does try to deal a lot with prevention and education as well as treatment. So now we get into kind of the last phase of the class. And this is one of my favorites. It's also, unfortunately, one of the more complicated ones because there's a lot of new terminology here, a lot of things you probably haven't heard of before. But ultimately, what we want to look at now is your body's defense mechanisms against infection. We spent a fair amount of time talking about the pathogen's offensive capabilities, particularly virulence factors. And when you look at an impressive pathogen like staph or strep, you kind of wonder, man, how can I ever survive the onslaught of that pathogen if I'm exposed to it? I hope one of the things that you come to appreciate just in this course is microorganisms are everywhere. And many of those organisms are potential pathogens. We're inundated by them. So why don't we get sick more often? Why don't we constantly have a state of disease? Well, think about what has to happen for disease to occur. You've got to be exposed to the organism. That's typically not a problem. Particularly when the weather starts getting bad, we start spending more time inside, and we start exposing one another to our germs more frequently. Just being exposed, however, does not mean you're going to have a disease. The bacteria have to establish themselves at some type of portal of entry. They've got to be able to attach, and then they've got to be able to invade the tissues and start spreading. Fortunately, we have a series of barriers that help to resist that onslaught. And what we're going to see is that we have several lines of defense that we usually break down and talk about as the first line of defense, the second line of defense, and the third line of defense that really are pretty effective in preventing us from getting infected most of the time. Now, sometimes are the barriers overcome? Absolutely. First line of defense are primarily mechanical barriers, things that really prevent entry of the pathogen or attachment of the pathogen. Things like your skin, your mucous membranes. But we're also going to see that there are some genetic components to this as well. There are some chemicals as well that are involved in this. The second line of defense also involves some chemical compounds. There are interferons. There's complement. There are a variety of chemicals associated with the process of inflammation. But there are cellular components involved in your second line of defense as well. And a big part of that cellular response involves phagocytosis. And also, oftentimes, a phenomenon called antigen presentation. So you're going to see me refer a lot to something I call just APC, an antigen presenting cell. So we'll talk about that a little bit. These first two lines of defense are ones you're born with. And they are typically referred to as the innate immune system. The third line of defense is a very complex interaction between various types of cells and chemical compounds. Many of the chemical compounds are a class of compounds called lymphokines. And they are involved in chemical signaling among the immune cells. The major players here are the antigen-presenting cells. They include macrophages and dendritic cells. Another major player are the B lymphocytes. Does anyone know what a B lymphocyte produces? B lymphocytes produce antibody. Antibody. And we'll see what antibodies are and how they protect you against infection. Another major player in the immune response are T lymphocytes. They don't secrete antibody. T lymphocytes either kill host cells or help to kill them. Why in the world would you ever want to kill one of your cells? If it had a virus or if it was mutated and or cancerous. Okay? Some cells in your body have gone bad. And what you're going to see is that T lymphocytes have an interesting interaction with B lymphocytes usually. They also react with these antigen-presenting cells. So the interactions between this third line of defense get really pretty complicated, but pretty interesting. This line of defense is called your adaptive defense. It's something you develop by being exposed to disease-causing agents. The adaptive defenses are characterized by memory, redundancy, and feedback. What's memory referred to? Once your immune system has been exposed to a pathogen, it remembers that encounter. And if you encounter again, your response is much more rapid and much more robust. That's why we vaccinate. We're trying to stimulate a memory response. Because the first time you see a pathogen, your response is pretty slow. It takes a while to develop. And if that is the real thing, you might die before your immune system gears up significantly enough to be able to deal with that infectious agent. If you've been immunized, that's typically not a problem. Syntax. What does redundancy mean? The American space program had a pretty impressive safety record, despite some of the shuttles, because one of its hallmarks was redundancy. Redundancy means you have backups for backups. Okay. There are built-in backups within the system. Unfortunately, the redundancy sometimes makes it difficult for you to wrap your head around, particularly this third line of defense. Because there are overlapping layers, if you like. The feedback that occurs within the immune system can both be positive and negative feedback. Positive feedback is kind of a self-amplifying process. The immune system basically activates itself to become more and more robust. And that's pretty important if your bacterial population is climbing during the log phase of population growth. Why do you need negative feedback? Yeah, you got to be able to shut it down at some point, right? And sometimes the body doesn't do that. And we run into chronic states of overimmunity. And that's as dangerous as not having immunity. So when we start showing some of these interactions in the third line of defense, the diagrams that I put on the board are going to get a little complicated. There are going to be all these arrows going back here and back there and stuff. That's really because of these particular aspects of the immune system. And also, it's a system that's evolved. And a lot of times, evolution is a little bit messy. Okay? And it's a little bit not as tidy as we would like it to be. One of the things that we'll talk about are, where in the body is your immune system located? What's your primary kind of reaction right now? Where do you think most of the immune reactions take place? Pardon me? Yeah, they actually do. Most people will say the blood. That's where they think all the immune reactions take place. But most of the significant immune reactions take place in the lymphatic system, including your lymph nodes. When you're exposed to a pathogen, a very interesting series of events take place where white blood cells, which are phagocytic, are patrolling around your body. They move by amoeboid motion, like an amoeba does, and they crawl throughout your tissues. And what are they doing? They're conducting surveillance, and they're looking for things that don't belong. How are they doing that? They're using their receptors to look for molecules that are deemed to be foreign as opposed to self. What happens if they detect something that is foreign? Oftentimes, what they'll do is they'll phagocytize it. And once they phagocytize it, they do disassemble it. But oftentimes, the process of disassembly also involves putting little pieces of that disassembled pathogen on the surface of your cell. That's called displaying the antigen. Why are they doing that? Because they want to be able to show it to other immune cells and activate those cells. How are those cells knowing that that stuff is out there? They have receptors that are looking for them. So one of the things that we're going to spend a fair amount of time on is talking about some of the receptors that various immune cells have and looking at those receptor interactions and what they trigger. And what do they trigger? Oftentimes, binding of a receptor triggers chemical signaling, getting other cells involved. And the chemical signaling may ultimately trigger antibody production or activation of these T cells, which are going to kill your body cells. We'll spend some time talking about the development of these immune cells. And that's a whole fascinating process in and of itself. For example, your body has the ability to respond and produce antibody against a completely synthesized novel antigen that's never been in the environment before. How can your body do that? How can your body respond to something that has been brand newly created? Bizarre. So let's start out by looking at the first line of defense, and that will transition quickly into the second line of defense, and then finally we'll put this together with the third line of defense. So some of the first couple of slides here are just kind of summaries of various parts of the immune system. This is kind of a silly illustration of kind of some of the things that are going on in lines 1, 2, and 3 of your defensive systems. It is. Should be. Line 1 and 2? No?
It's just the notes.
I will have Natalie add it. First line of defense really has three basic levels, or three basic components. There are physical barriers, some chemical compounds oftentimes associated with those barriers, which are typically tissues. And then there are some genetic components associated with those. Really, the goal of the first line is to kind of prevent entrance or attachment. Primary locations that we have, the first line of defense, are the skin, excuse me, and the mucous membranes. Now, some of the information I've already gone over when we talked about the four tissues of the body. And we talked about epithelial tissue, both keratinized epithelium and epithelium mucosa. Skin cells have a number of adaptations that make them a good barrier. And really, unless this barrier is breached somehow, there's a cut or a scratch, it is a very effective method of preventing bacteria from gaining entrance into the deeper tissues. That's why burns are so devastating, particularly if you're burned over a large part of your body. Because that protective outer barrier has been compromised, and it's just really difficult to get those infectious agents under control. Skin cells secrete collagen. That makes the skin more pliable. When skin gets too brittle and dried out, it cracks more easily, and that would cause compromise of that particular barrier. The skin can produce antimicrobial proteins that are called defensins. And those can have a variety of actions in terms of breaking down pathogens. There are populations of dendritic cells patrolling throughout your skin that are phagocytic. The skin itself does have some weaknesses. When we talked about acne, we said that the pores of the skin are potential sources of entry for bacteria like Staph aureus. And when they start to colonize then a hair follicle, for example, that they can cause inflammation and can potentially penetrate into the deeper tissues. Associated with the skin are sebaceous glands, and they produce sebum. Sebum has fatty acids, and that will create a small microenvironment of lower pH, which tends to retard microbial growth. The outpouring of sweat can cause a flushing action in the skin and help wash pathogens out. Sweat is usually a little bit salty. And that high osmotic environment can retard microbial growth of a lot of things. Unfortunately, not staph. While it's not the primary place that this is produced, skin cells can also produce lysozyme. Remember what lysozyme does? And don't confuse it with lysosome. Remember, lysozyme degrades peptidoglycan. Where else in your body's system of defense do you find a lot of lysozyme production? Pardon? Not kidneys. Your saliva and tears. The mouth is very prone to infectious agents. I think you put in your mouth. And the eyes are constantly exposed to dust and dirt particles. So lysozyme is a pretty effective chemical that can help to prevent infections in those particular areas. We talked about the epithelial mucosa, and particularly the mucus that's secreted that can trap pathogens, prevent them from attaching. The cilia associated with a lot of mucosa, which can create that sweeping motion that sweeps things out. The smooth muscle associated with your digestive tract is peristaltic, and the rhythmic contraction of the smooth muscle can move things through the small intestine and the large intestine. What's one of your body's typical responses to infection? Increase peristalsis or slow it down? Increases it. It tries to get rid of things. What is the associated symptom associated with increased peristalsis? Diarrhea. Okay. Body fluid spends less time in the large intestine, less reabsorption of the fluid, and so diarrhea can result. So should you typically take anti-diarrheal medicines? You might be instances where it's really indicated if your diarrhea is just really horribly out of control and dehydration is a problem. But if you take anti-diarrheals like Imodium, that slows down the peristalsis and really compromises one of those particular major defense systems. Stomach acid is another important part of your body's first line of defense. Because remember that the basically tubes that go through your body, including your esophagus and your intestines, are considered external, and you ingest or inhale a lot of pathogens. If the food that you eat immediately is dropped into a vat of acid, that promotes a little bit of digestion, but the major function of the acid is destruction of microorganisms. So when things enter the small intestine, hopefully they aren't still viable. I mentioned that iron is a very important nutrient for microbial growth. And oftentimes it is a limiting factor to microbial growth. Iron really isn't very soluble in the blood. So our body produces a protein which binds iron and helps to move it in the blood called transferrin. Staph aureus has developed a strategy where it produces a protein called a iodophore. And basically it competes with the body's transferrin for iron and tries to grab the iron away from it. So there's kind of a tug of war for iron that can be going on in the blood between the pathogen and the body. Some bacteria bypass that strategy and produce enzymes that cause red blood cell lysis, which can liberate the iron. Excuse me. So these are an example of some of the kind of general components associated with your body's first line of defense. And really, pretty effective. There also are, though, some genetic considerations that you've got to think about when you talk about the body's first line of defense. There are certainly very defined racial and ethnic susceptibilities to certain types of diseases. Smallpox originated in Europe. It was not really found in the New World until Europeans colonized the New World. So people of Native American ancestry were very susceptible to smallpox. Okay? Natural selection had not yet worked on that population to kind of weed out the individuals that were most susceptible, as they had more in European populations. Unfortunately, this particular susceptibility, according to documents, resulted in some of the first biological warfare that was conducted in the history of warfare. And there are well-documented accounts of the Army passing out blankets infected with smallpox spores to Native American Indian populations, creating smallpox epidemics. What is the basis of these differences in susceptibilities? Receptors for the virus. Okay? If the virus cannot attach to your cells, it cannot infect them. Over a period of time, a certain amount of resistance will develop to diseases like smallpox as random mutation changes those particular receptors. The natural selection would favor the survival of those individuals with those mutated receptors. So we can see the evolution of resistance over time. Another interesting genetic consideration is that some genes are pleiotropic. Remember from genetics what that term means?
Is that one of the
Yeah, kind of. It's really that a gene that can affect more than one trait. The classic example is the sickle cell gene. If you have 2 copies of that particular gene, you have a debilitating disease called sickle cell anemia. But that gene, which is pleiotropic, also influences what other factor? What if you're heterozygous for the sickle cell gene? Malaria resistance. Okay? Sometimes a single gene difference can have a devastating effect on your immune system. There is a single gene defect in a protein called adenosine deaminase. And this is a crucial gene that, if it's defective, interferes with normal lymphocyte development, B and T lymphocytes. Individuals who suffer from this particular genetic defect suffer from a condition called SCIDS. That stands for Severe Combined Immunodeficiency Syndrome. And basically, people who are born with this have neither B nor T lymphocytes, functionally giving you really no immune system. If they can diagnose this through genetic testing in utero, what are the options? Best case scenario would be correct a gene defect in utero. And there was research that was done with that. There were some cases of success. But the vector that was used to deliver the gene was a virus. And they really had to shut down the trials, and they've never been restarted again because people were dying from cancer caused by these particular vector viruses. What's the other alternative? Deliver the children into a plastic bubble, basically. There was a story a long time ago on TV. A boy in the plastic bubble. And he lived his whole life. Could not be exposed to any outside air because he had no immune system. So that single gene defect can have that devastating effect on immunity. Well, what we're going to do next time is talk a little bit about the structural kind of breakdown of how the immune system is organized. And we're going to talk about it in terms of 4 compartments that you find in the body, which are the primary locations of the immune reactions. Those 4 compartments are your extracellular fluids, a system called the reticuloendothelial system, which you've probably never heard of before, the lymphatic system, and the blood. And when we get into the blood, we will quickly transition into a fairly detailed discussion of the major cellular components of the blood, particularly the white blood cells, and give you some understanding of what those different white blood cells are, what their roles are in immunity, and how you can tell them apart from one another by particular staining methods. So wherever we finish up tomorrow, or a convenient place that I'll tell you, excuse me, when we get there, will then be the breaking point for the exam. So I'll go back now and make sure that I get the PowerPoint for this on Dropbox. You said the lecture notes are there? Okay.